This is the third in a series of 3 videos on the topic agonist efficiency. In the first video, I talked about the receptor, basics of receptor-agonist interaction. And in the second, I described the concept of efficiency, which is the correlation between efficacy and affinity. And now in this third video, I'm going to be talking about the energy linkage that undergird that correlation. And the specific topics I'll talk about are agonist binding, some energy correlations that are apparent in the data, just simply observations. I'll talk about intermediate states in binding and gating. And finally, I'll put it all together and propose a linear free energy zipper mechanism which unifies binding and gating. So the central question that we want to answer is, why do agonists with very different affinities and efficacies have the same efficiency? This was discussed in the last video. There is just an observation, empirical observation, that there's a correlation between the low and high affinity equilibrium dissociation constants on a log-log scale, and many agonists fall on this line independent of their efficacy and their affinity. And that relationship relates to efficiency, which is defined as 1 minus the log equilibrium dissociation constant ratio. So the question is, what is the mechanism that determines that all these agonists have the same binding energy ratio? So to understand this, we have to talk a little more about agonist binding. Agonist binding to nicotinic acetylcholine receptors is not by simple diffusion. So these values are the on rate and the off rate, the association and dissociation rate constants for 4 different agonists to an individual binding site. I think this is the alpha delta site in both the C and to the O conformation. So we made channels in C, we just took channels and added the agonist. For O, we made channels activate constitutively and then add agonist, and we could detect the transition to the liganded O state from the unliganded O state. So O is for orange is for O, and cyan is for C, closed channels. And by simple inspection, you can see that C is less than diffusion and agonist dependent. So all of these agonists have approximately the same diffusion constant, and there's little difference in their rotation. TMA has no rotation. It's a perfectly symmetrical molecule. So C is slower than diffusion and agonist dependent. As far as dissociation goes to the right, to C, they're all approximately the same, about 10,000, 20,000 per second. But to O, they are slower and more agonist dependent. So that is an observed, an empirical observation that was first looked into in 2001 by Claudio Grossman and shown in the citation down below, but analyzed in more detail in 2017. So this is, I think, a very robust finding for these 4 agonists at 3 different kinds of binding sites. So the question is, how do these results impinge on some common misconceptions about or conceptions about agonist binding? I think many people would say agonists diffuse into their target site. And you can see, as I said before, binding into the C target site, the binding is slower than diffusion and agonist dependent, even though the diffusion constant is the same. So that is definitely not the case for entering C, possibly to O. O is closer to diffusion and less agonist dependent. So agonists do not diffuse into the closed binding site of nicotinic acetylcholine receptors. And this observation of slower than diffusion and agonist dependent is repeated in GABA receptors and glutamate receptors, other receptors where it's been measured. So the second conception about people have about agonist binding is that the higher affinity of O is caused by a steric barrier. People imagine that a loop closes over the binding site, or there's a clamshell motor, something sterically closes over the binding site to trap it in there, like putting a lid on a jar. And that's why it's high affinity. There's a steric barrier, it can't get out. And if you look at dissociation, that could be. The off rate from the open channel is slower than to the on rate. and is slower than to the closed channel, so that's consistent with a lid being in place. However, the association rate constant data is inconsistent with that hypothesis. Getting into C is slower than getting into O. K on to O is faster than to C. If there is a lid in O, then it should not be able to get in there faster. The barrier that we're thinking about must pertain to C. There is a barrier that is removed when the channel goes from C to O. Now, I don't know if it's a steric barrier, it could be a tiny barrier, it could be a small charge change, or just a rotation of a single bond. I don't know what that barrier is structurally, but agonists can't get into the C binding site, but they can get into the O binding site by diffusion. And the third conception that people have is that it's possible to compute affinity by docking. So you can dock molecules into structural models of the binding site and calculate the binding energy, which gives you the stability of the closed state, which relates to the off rate constant. That stability determines the off rate constant. But this cannot be used for acetylcholine receptors, GABA receptors, glutamate receptors, because KDC, the equilibrium to constant to the closed state, is set by K on. On the right-hand side is that for C, the blue bars for closed channels are approximately the same for all agonists. So you can dock away, but the off rate stays the same for all these different agonists. So I think that may work for other receptors and other agonists, but not for the ones listed down there. So I want to continue with this idea that KDC is set by K on and discuss, take a little sidebar here and talk about correlations. So there are three agonist energy correlations. These are experimentally observed facts. These are observations that need to be incorporated into any conception about how the acetylcholine receptors work. So let's do the first one is the one we just described, which is the KDC is determined by K on. If you make this plot, which is a log-log plot of the rate versus equilibrium constant, that's called a rate equilibrium free energy relationship. This is for one binding site and four agonists. You can see that the slope of that line is 1, meaning that the on rate is completely determined the equilibrium constant. The off rate is the same for all agonists. So phi is the slope of that relationship, and B stands for binding of the agonist. So these are all pertain to the agonist. So the phi value for binding of the agonist is 1. That means that the on rate is what varies between agonists. The off rate stays the same. So that's one strong agonist correlation. The second is one that we discussed in the first video, which is the phi value of the agonist in the gating reaction. So this one, you plot the log of the opening rate constant versus the log of the gating equilibrium constant. This is with a fully liganded receptors, and you can see for all these agonists, including water, falls on this line. The opening rate is what determines, or largely determines, the equilibrium constant. Various measures are between 0.93 and 1 for this. So I took the value of 1 to keep it simple. So that's another rate equilibrium free energy relationship for gating. So we have phi binding and phi gating. And we also know now from the second video is that there's a correlation between these two equilibrium constants. So this is not a rate equilibrium free energy relationship. This is basically an equilibrium free energy relationship. And efficiency is 1 minus the slope of that line. So we know phi binding, phi gating, and efficiency for agonist families. And we also know there are multiple agonist families. I've just shown one here. So we can use these three correlations to simplify our analysis of receptor activation. So here's a standard model for receptor activation, sequential model now, where you bind n times until you form a fully liganded receptor, which then undergoes the isomerization from the inactive to the inactive form, C to O. That's called gating. And this is used over and over again for many different agonists. Of course, this is really a cyclic model in which each of the intermediate ligation states can also undergo the isomerization, but those events are rare in most receptors, so they can be ignored. So this is the main sequence of events that gives you a response, gives you your current. So the three correlations I showed on the last slide, plus two receptor constants that are true for all agonists, plus one experimental measurement can give you the response. And I'll show that, I'll just break that down in this slide. So let's say the experiment, we know N is the number of binding sites. So let's say that's a given. And we make one experimental measurement, which is K on. I don't know how you measure this. You can measure this any way you want, with a biocore, with a patch clamp, any number of ways. You measure the on rate constant. Now, because of phi binding, you know K off is the same for all agonists, so you know KDC for that ligand. And now, furthermore, since you know the efficiency of that ligand, as I discussed in the last slide, that could be deduced from structure perhaps. So you know phi binding, you know the efficiency, you now know KDO. So you know KDC and KDO, given phi binding and the efficiency. And now that with N, if you know L0, then you can use the green equation from the first lecture, which is from the thermodynamic cycle. You know that Ln is equal to L0 times the coupling constant, which is KDC over KDO raised to the nth power. So we know that equation, so now we know Ln. And furthermore, since we know phi gating, we now know the opening and closing rate constants. So from one experimental measurements, we've calculated all the rate and equilibrium constants for every step in this scheme. We can combine, we can take either KDC or Ln, since we know efficiency, those are related. I described that at the end of the second lecture, to calculate the dose response curve. And since we know the rate constants, we can also calculate this is the end plate current or the synaptic current decay, the shape of the synaptic current. So from one experimental measurements, we can calculate two very important functions. So if I was a drug company executive, if I owned a drug company, I would say, given a receptor, measure its L0, given some agonists, measure their efficiencies to determine the agonist families, and then take a billion compounds and measure K on for those billion compounds. And assuming values for K binding and K gating and efficiency, which you have to measure, those numbers, assuming those three numbers, once you get all that, and L0 and the number of binding sites, you can calculate your dose response curve. So you don't have to grind through all that, all those data. You can calculate it from here. Of course, you need to know desensitization and things like that as well to get a cellular response. But this is just for the binding and gating parts. Okay, let's get back to efficiency now and the mechanism that undergirds efficiency. In 1913, Michaelis and Menton proposed an intermediate state in the conversion of substrate to product and enzyme, namely the ES bound substrate state. And that was confirmed 30 years later by Britton Chance after he had invented a stop flow apparatus and was able to detect this state directly. By analogy, in pharmacology, in 1955, Del Castillo and Katz proposed an intermediate state in the activation of receptors, namely one where the agonist was bound but the channel was still inactive or closed. And that state was not detected directly until 1985 by Cahoon and Sockman, after Nair and Sockman had invented the patch clamp. So there are undetected intermediate states inside of chemical reactions that later on become detected. So likewise, in 1992, I detected a short-lived shut state that I put as an intermediate state between AC and AO. That's a figure from the paper. And it was a difficult detection, and it was not very convincing. And in 2011, the University College London group and the Mayo Clinic group both detected the same event, and they named it flip or prime, respectively. I'll refer to it as flip, but it's the same as the stepwise event that was detected in 1992. So there are always undetected intermediate states, in these complex chemical reactions. And I think the data I just described about agonist binding suggests that the binding process is not simply diffusion. There is a diffusional component, but there is also a conformational change that removes that barrier to allow the agonist to bind with low affinity. That's the blue arrow. There's an intermediate state somewhere in the binding reaction, and I will go on to say there are many intermediate states in the second step of that process, the green arrow, for gating. So let's look at those two steps in more detail. So there are two agonist-dependent rearrangements of the binding site. In binding, there is a diffusional process and that conformational change that allows the formation of the low affinity construct, and that is shown as a gray bar for diffusion and a blue bar for that conformational change. And the reason the gray bars are pretty much the same for all the agonists that we talk about, but the blue bar is the part that's agonist dependent. So KD, the agonist dependent component of KD, is determined by the blue part, that conformational change. So the two of them together combine to give you KD, but the agonist part is in blue. And similarly in gating, I'll go on to show you that there are, I think, 4 intermediate states. That AC prime state or flip state really is composed of 4 different high affinity non-conducting states. And together these states form a gating conformational wave that connects the binding site with the gate region in channel opening and reverse in channel closing. And again, the only part of this wave that is agonist dependent is the first step, which determines the coupling constant. That's the switch between the low affinity to high affinity takes place, and that's shown as that green bar in there. And I'll describe this in more detail below, but I'm going to name these two colored processes. The blue square that's involved in forming the low affinity complex is called catch, and the green rectangle that's used to that signifies the change from low to high affinity I will call hold. So in catch, the agonist enters the pocket and binds with low affinity. That's determining the energy in on the dose response curve. And in the green rectangle in hold, the pocket contracts. That's our hypothesis. Described in the second video lecture. The binding energy increases to determine the efficacy, which is energy out. So affinity is set by something by catch, which takes place in binding if we work back up the chain, and efficacy is set by hold that takes place in gating. So we've turned binding into catch and gating into hold when we're talking specifically about the agonist-dependent energy changes. So this idea of the 5 states was proposed in the first part of this century, from about 2000 to 2013 is when we worked out those 5 different steps. But the data that I showed before in the last video lecture with the efficiency plot, which is a plot of log gating versus log low affinity binding on those axes, that correlation, which is efficiency, is really reflects catch and hold. So low affinity binding is reflecting the catch rearrangement, and the low affinity and the gating equilibrium constant, the agonist dependent component of it, is reflecting the hold equilibrium constant. So catch and hold energy changes are correlated. That's what efficiency means. Something in binding, the energy change in binding is correlated with something in gating, and that something is catch and hold. So why do agonists with very different affinities and efficacies have the same efficiency? So the question is, why are all those agonists fall on the same correlation line? Binding and gating are 2 different processes, but we now know catch and hold are 2 different processes. So let's break this down in terms of a energy landscape. So we see, I'm now showing just the colored bars, the blue and the green part of the energy landscape. There is a energy change associated with diffusion of the agonist, and there's an energy change associated with the rest of the gating conformational wave, but those are not agonist dependent. So we're just going to focus on catch and hold. And the idea is that the energy change in catch and the energy change in hold are related by a linear free energy relationship. So that's shown as these two humps. The humps are the transition states for catch and the transition states for hold. And you can see that the energy change in the first hump, which is catch, is log KDC, proportional to log KDC. And the energy change in the second step is proportional to the coupling constant energy change, which is log KDO minus log KDC. So the overall energy change for catch and hold is the sum of those two, which is log KDO, and the fraction of that overall energy change that is devoted to the gating part, which is hold, is shown as 1 minus the log of the two, shown in the pink equation to the right. So this is another way to derive the equation for efficiency. The fraction of the overall binding energy that is devoted to hold, which is gating, is that equation. So what this means is the key is that those catch and hold are tethered by that dashed line, which is the linear free energy relationship. That's the basic idea. And the other idea is when you have a weak agonist, the tilt of the overall energy landscape is less. That's the red line for a weak agonist. But the fractional changes in catch and in hold relative to the total remain constant. So even though the overall energy is less for a weak agonist compared to a strong agonist, the fractional changes are the same, and therefore the efficiency stays the same. So that's the basic answer to the question. Why do agonists of very different affinities and efficacies have the same efficiency? It's because the catch and hold rearrangements are linked by a linear free energy relationship. So what that means, the implication of that is profound. That means binding and gating are linked energetically. So that means there is crosstalk between energetic crosstalk between what happens in catch, what happens at hold. And in this slide, I'm going to go through that in detail just so the idea is clear. So the idea is that the agonist shows up, and what the presence of the agonist outside of the binding site, before it gets in to form the complex, it lowers that energy barrier to get in, the catch energy barrier, and it lowers the catch state. So those two red arrows are caused by the appearance of an agonist at some site outside the binding site. I don't know what the site is. It would be important to find out. I don't know what the conformational change is. It would be important to find out. But this is what the data, the kinetic data, is telling us, that agonists cause a change in the entry of the change in the catch, promote the catch conformational change. Now, perhaps the agonist resembles the transition state of the catch rearrangement, and that's why it promotes it. I don't know. But the agonist promotes catch. And then once catch happens, these are the energy landscape after catch but before hold is the gray line. After that happens, so catch happens, and then after catch happens, that event causes a stabilization of the transition state and the end state for the hold reaction. Those are the blue downward arrows. And then hold happens. So agonists promote catch and catch promotes hold. And that's what that dashed line means, why that means they are tethered together by a linear free energy relationship. So that is the basis of efficiency. The catch energy in and the catch energy and the hold energy out are connected through that linear free energy relationship, and that's why there is such a thing as efficiency. That's why agonists have the same, a group of agonists has the same efficiency. I'm going to show this one other way to make it even clearer, and that's as a reaction scheme. So the main activation pathway is the vertical lines are diffusion of the agonists to form the A-C complex. And then the blue lines represent catch, and the green lines represent hold. So the main activation sequence is diffuse, catch, hold. Now it is possible to hold before catching, and that would be on the bottom plane, that would be the leftmost green arrow. But the arrow thickness represents the equilibrium constant for that reaction. So because of the energy coupling between the two events, the rightmost green arrow, which is hold after catch, is much higher equilibrium constant than the leftmost arrow, which is hold before catch. But because you have to balance the energy around the cycle down below, that if there's a small arrow going from AC of hold before catch on the left, then the catch arrow must be large in order to balance the two main pathways. So here's the main pathway, diffuse, catch, hold. And now you can see that catch promotes hold, as I described, but also hold promotes catch. So if somehow you can get into the hold without catch conformation, then catch is going to happen very quickly. And this is going to be important when we describe the zipper mechanism at the end. So I hope that's clear. There's crosstalk between binding and gating energy structural changes, and that crosstalk is what is the answer to the question why many agonists have the same efficiency. So let's talk about the conformational wave. So this comes from a large series of experiments carried out in basically between the first decade of the 21st century that we did, which is measured thousands of mutations. And if you measure the effect of a mutation on the rate and equilibrium constant for gating, that relationship is called phi. I described it to you as a correlation of phi gating is equal to 1 for the agonist, but there are other regions of the protein that can be probed besides where the agonist binds, and by making mutations. So these are all come from mutations, and what they are is the ratio of the two vertical colored bars in the energy landscape shown in the inset. So that's the change in the energy of the transition state relative to that of the end state. And that gives you this parameter called phi, and that's determined by fitting a series of mutations at one residue. So the slope of that line, just like the slope for the agonist is 1, we measure that slope for a series of mutations at a different residue. And we've probably probed 3 or 4 mutations in about 100 different binding residues, something like that, to get this following histogram. So all these phi values, again, they're not spread all over the place. They fall into discrete populations. The population values are shown above, 0.95, and that is mapped to mutations of residues near the binding site. So those aromatic residues I described before are 0.95. If you move away from there and you go into the extracellular domain, those are the red ones. The phi value drops a little bit. It drops to 0.8. This is mostly in the alpha subunit. If you move further away into the transmembrane domain, that's in yellow, shown to the right, the phi values of mutations there are around 0.58 or 0.6. If you go in the area around the gate, you get low phi values of about 0.3. And if you go exactly at the gate, which is, the 9' residue of M2, you get a very low phi, which is 0.06. So we see 5 different populations that are located spatially. There are some exceptions. There are some high phi values somewhere down in the interface between the red and the yellow. So, but this is in general the map of so you get from this phi analysis, you get spatial information, you get a map. You don't get temporal information, you just know the ratio of the energies, but you do get spatial information. And we developed a model, a theoretical model, to show that phi values can be related to a position in a linear reaction sequence. So if the different domains shown at the right switch from C to O in sequence, then the phi values gives the position of those sequences. So there are 5 phi values, which means 5 transition states, which means 4 intermediate states between AC and AO. And the high affinity change takes place in the green step, the hold step. So everything after the end of hold is high affinity. So I've named these in the spirit of flip. I've given these names that somehow relate to the functional process or the structural or functional process that takes place. Hold is the contraction of the binding site. Twist, there is a small rotation of the extracellular domain in other ligand-gated channels that resemble the nicotinic receptor. There's also a contraction of that pocket, but it's called blooming, unblooming, but it's a little clunky to say that. So I'm just going to call it twist. The membrane helices rearranged, and that's called tilt. And rip, I call what's changing in the circumgate area, the residues near the gate. And pop is when the water compartments merge. So the idea is that there's a hydrophobic switch at the gate region, and a change in small change in the side chains there leads to the ability of water to enter and thereby join the two compartments and allow ions to flow. So there's a time when there's sort of a bubble or a region of low water density there, and pop is the event which eliminates that bubble. So down below this reaction scheme, I've given the locations. So when the channel opens, it goes through these in sequence, just like catch and hold were in that cubic model I showed previously. You would have hold, twist, tilt, rip, pop into a six-dimensional model. I can't show the model; it's too complicated to show because everything can happen. It's possible that tilt happens before twist, for example, but it's unlikely. That this is the most likely sequence of events: is the linear chain going hold, twist, tilt, rip, pop, going down, and pop, rip, tilt, twist, hold when the channel closes. So this is the conformational wave, because you see on the right, it starts at the binding site, then it goes to the extracellular domain, then in the opening process, and then goes to the membrane domain, and then to the gate, and then to water. So that's the idea of the gating conformational wave. And again, this says there are 4 high affinity pre-open states. Again, this is proposed in 2005, let's say, and it's going to take 30 years before this is found. So maybe in another 10, 15 years, people will start finding these intermediate states in structures. But right now, they are coming from kinetic analysis to 5-value analysis. And the other thing that I can say is we don't know the time from this phi analysis, but the direct detection of flip and prime is useful in that it gives us a time axis for all these events. So flip, which is shown there under pop, is on the order of a 5 microsecond event, and that minimally represents the collapse of the formation of the gate, the bubble at the gate, because it's starting from O. You see a closure, but it could represent any number of steps going all the way back to the hold event. The whole thing may be involved with what the flip event is showing. The patch clamp is a great instrument, but it really is limited in terms of bandwidth when we're talking about conformational changes in proteins. It's been estimated, we estimate that these things are each of these steps takes on the order of 100 nanoseconds or so, which is well beyond the time resolution of the patch clamp. And I also want to note that I have named these events not by the state, but I've named it by the transition state, which is the rearrangement, the dynamic event that is represented by these energy barriers. So hold refers to an event, flip actually refers to a state. So there's a small difference in the naming system there. So let's put this together. The idea is we're going to extend that linear free energy linkage between binding and gating, between catch and hold, to all the other events inside of the gating event. So what's shown here is the energy bandwidths, which are the low resolution patch clamp, the 5 microsecond whatever resolution, or 20 microsecond resolution patch clamp shows us there's only a C and an O state with some energy barrier between, and that the agonist causes an uphill reaction to become a downhill reaction. These are potential energy surfaces. Now I'm going to show these surfaces, imaginary surfaces, for all the gating conformational wave. So the blue line is without agonist. There's an uphill catch reaction, and then there's another uphill cold reaction, and then there's all the other steps. And these are sort of arbitrary amplitudes. We don't really have much information about what's going on inside of the conformational wave energetically. But the idea is that after diffusion, the agonist lowers the barrier and the end state for catch, which in turn lowers the barrier and end state for hold, which in turn does the same for twist and then tilt and then rip and then pop until the whole thing comes downhill. So the agonist triggers catch, which then triggers hold, blah, 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 all the way down to open. And if you took this blue line in the expanded version and condensed all those barriers together because you didn't have the time resolution to see them, you would see the blue barrier that's shown in the inset. That's how you would model it. And similarly, you would show the black line if you didn't have the bandwidth to see all the barriers, and it would show up as a reduction in both the transition state energy and the end state energy, as I described in the very first set of slides in the first lecture, that the agonist lowers the transition state, and it does so because hold takes place at the beginning of this reaction chain. And that's why the transition state goes down, because hold is the first step that happens. And I think in voltage-gated channels, S4 motion is the first thing that happens. So if you have a perturbation like voltage or a stimulus that increases both the rate and equilibrium constant, then the region of the protein, the sensor region for that stimulus, is going to change its energy from resting to active at the beginning of this chain of events. So this explains two results. It explains the lowering of the transition state energy and therefore the fast activation, as I just described. But it also describes that weird result that the binding to C is not by diffusion, whereas binding to O has. So that means binding to O is diffusional, means that catch is already there when you cause the channel to open constitutively. So the right red arrow means I've made a mutation that lowers the state, lowers O, that forces the system into O. So that means everything is going to be propagated backwards, as I described in the cubic model, so that, you know, not only does tilt promote rip, but rip promotes tilt. So when you fuse the water molecules by pop, then you will promote the rip event, and rip will promote the tilt event. Tilt promotes twist, twist promotes hold, hold promotes catch. So catch is happening in the receptors that are made constitutively active by making a mutant. So that's the idea. I call this the zipper model because it looks like a zipper going from one energy landscape to the other, because each notch in the zipper promotes the next notch in the zipper. And same as unzipping, going backwards. So you're zipping and unzipping the conformational change in this model. So I call this the zipper model of acetylcholine receptor gating. So let's do one more simulation. I'm going to show this zipper mechanism as an animation 3 times. The first time you should be looking at the energy landscape. The inset shows the low bandwidth recordings, and the main landscapes are for the individual steps. So the 3 components of the slide are the landscapes, the structure, and down below the reaction scheme. So I'll show this 3 times, and we can look at each of these 3 components at each step, each slide. So first I'm going to show the energy landscape changing in stepwise fashion as it goes from unliganded closed to liganded open. So the blue line is unliganded uphill reaction. Agonist shows up. So each of those steps was arbitrary. There's some indication that the that the twist event is the most energetically difficult of these steps along the process, around the activation process. So now I'm going to show it again. This time we can look at the reaction scheme down below to see what each of these uh, intermediate. So in the second animation, let's focus on the reaction scheme down below and the four intermediate high affinity pre-open states and the events that are taking place. First is diffusion, then catch. Hold forms the first high intermediate, flip intermediate, twist is the second, after tilt is the third, complete is the fourth, and then finally you get to open. So there are four high affinity closed states in this model. And now in the last animation, let's focus on the structures, which is one of the beautiful benefits of the phi analysis. It may not give you time, but it does give you structural location. So here we go again. This time let's look at the protein to the right. That's the ion up above. Agonist shows up. Catch, hold, then the extracellular domain twists, then there's the tilt of the helices, rip, and then pop. So that's my idea of, this came from efficiency, that the correlation between catch and hold is the essence of efficiency, and that linear relationship can be propagated down the line all the way to open to give you the zippering and unzippering of the acetylcholine receptor as its activation. And in this model, the distinction between binding and gating has been blurred. The energy, because there's what happens in a binding event is energetically linked to something happening in a gating event, means that binding and gating are really sort of an arbitrary distinction, that energetically this is one great big sweep from diffusion to ion flow. So with that, I want to give some acknowledgments and acknowledge my collaborators. In efficiency, the key observations were made by Snehal Jeti and Prasad Purohit, and was followed up magnificently by Tapan Nayak, Eva Bruhova, Rahima Bij, and now Dinesh Indurthi in my lab is working mostly on the whole cell aspect of measuring efficiency, and is interested in extending this to other ion channels, other receptors. The conformational wave took up the bulk of my middle of my career. It started with Claudio Grossman when we noticed started to build the map of phi values. But Ming Zhou and Claudio were on the initial paper. Yu Zhou worked out the model, the linear model relating it to phi values, and all these other people, Sudha Chakrapani, Alanya Mitra, Shweta Gupta, Giselle Simis, Archana Jha, Stuart Lick, Pallavi Vapna, Dave Kadugan, Jian Chen, and Wayne Sigurdson all were involved in some aspect of measuring the binding and gating constants that led to the wave. The MD simulations were first done by Sushri Tripathi and Sri Rupa Chakraborty under the direction of Wenjin Zhang in the physics department, who was my collaborator in all of the MD aspects. And now I'm working with Jay Singh and Pooja Goyal to try to understand the changes, the structural changes at the binding site, and relate those to efficiency. Desensitization work was done by Frank Salamone, Sergio Elenis, and Gustav Ach. I didn't have a chance to talk about that. That would possibly be another video lecture. And Gabriela Popescu, Louis Premkumar, Youngling Zhu worked on NMDA receptors, and Gareth Lima worked on GABA receptors, which contributed to our overall thinking, but not to the details of what I presented here. I've had several important consultants in this work. John Richard and Rob Colson are chemists, and John turned me on to the linear free energy relationships. He was a student of Alan Furst, who was a big proponent of using those. They're also called Bronsted plots in the chemistry literature. And Rob Colson gave me the idea that the binding and gating were linked by a linear free energy relationship, that tilt of the overall landscape. Eric Auerbach is my son, and he's a civil engineer, and he told me one day at Thanksgiving that an energy relationship could reflect efficiency. So for many years, we knew there was an energy relationship. The low and high affinity binding energies were correlated linearly, but didn't know what it was mean. And it was after that Thanksgiving dinner that I went back to my thermodynamics books and realized that what that meant was efficiency. So that was a big conceptual breakthrough. Uh, Fred Sachs and Feng Chen not only have been the brilliant minds that I've consulted with over the years, but they also were responsible for the software that allows us to analyze the data that we have. And they deserve great credit for writing both the algorithms and the code and the ideas behind that software. Pablo Paez let us use his ion flux machine. Timothy Gregg at Canisius College, chemist who helped us build molecules, synthesize molecules. Jolly Gao is another computational chemist who used to be at Buffalo, and I started working with him on molecular and quantum mechanical calculations of the binding site. And Andy Harris is a friend and colleague who made me think hard about that linkage between binding and gating, between catch and hold. Finally, I've had outstanding STEC sports that's been very stable throughout my career. Jan Jordan's been with me for 40 years. Mary Teeling also for 40 years. Very solid, excellent, outstanding technical support. Chris Nicolai wrote wrote a lot of the software for that was designed by Fred and Feng. Mary Merritt Bailey were molecular biology technician. Marlene Sharrow for long-term molecular biology, to making all those thousands of mutations that are, and if anybody wants a mutation of the acetylcholine receptor, please let me know. We have thousands of them in our freezer. And finally, I want to thank the funding agencies. NIH has been very good to me over the years, although not so much lately when it comes to efficiency, but for all the conformational wave, they were very enthusiastic supporters of that research. And SUNY Buffalo for paying my salary and giving me an excellent lab to work in. And I thank you.